it is coincided with the Teachers' Day. Every teacher is the role model for young minds and builds the nations with their strengths and with their inspirations. Good evening, dear Sister Nirmal Jyoti, patron and secretary of Nirmala College of Pharmacy, Dr. S.K. Abdul Rahman, principal and convener, Dr. D. Vinay Kumar, the coordinator of AICT ATAL sponsored PDF program, FDP program, faculty members of Department of Pharmacology and Pharmacy Practices of Nirmala College of Pharmacy, and all research scholars, delegates of renowned universities and pharmacy colleges who are registered with required skills and knowledge in the selected topics. My hearty congratulations for organizing faculty development program on the novel and contemporary research in the clinical research and clinical trials for 2 weeks, both online and offline mode. I feel honored to acknowledge and appreciate the hard work of correspondent, principal, HODs, and faculty members of the Nirmala College of Pharmacy, because in the first phase, our college is privileged to have this grant from AICT ATAL for faculty development program. I congratulate and appreciate all the participating faculty members of different colleges for what you do to the students and society for their overall development in adopting new ways, especially in clinical research and clinical trials through this program. These development programs and assessment can join force to improve student learning. If one faculty is equipped with novel and contemporary research, thousands and lakhs of students are enlightened and guided to serve the society with their new and renewed researches and approaches towards clinical field. This faculty development program that you have planned definitely helps you richly. I am also proud and immensely satisfied for the committed members of the family of Nirmala College of Pharmacy for conducting such training programs in the past successfully and effectively. Who dares to reach must never cease to learn. I wish and pray that this faculty development program will be completed successfully. My blessings to you all. Thank you, one and all.
Thank you very much, sister, for your time and blessings for us. We are grateful to listen to your kind words on all of us. Thank you very much, sister. Now we have the patron of today's session, very kind-hearted, Reverend Sister G. Nirmala Jyoti Garu, Secretary and Correspondent of Nirmala College of Pharmacy, to deliver our blessings and message for the delegates. Hearty welcome to you, sister.
A very good evening to all, especially the Chief Patron, Reverend Sister K. Maria Sundari, the convener and the principal of Dr. S.K. Abdul Rahman of Nirmala College of Pharmacy, and the coordinator, Dr. T. Vinay Kumar, the professor of Nirmala College of Pharmacy, and the co-coordinator, Dr. Shreya, of AICT Training and Learning Academy sponsored FDP program on the novel and contemporary research in clinical research and in clinical trials. I am happy to acknowledge that it is an appreciable and the noble task on the part of yours to obtain the grant from ATAL Academy, where it is providing a variety of opportunities for training and exchange of experiences through teaching and learning progression. I congratulate at this gesture for your significant efforts by fulfilling the objectives of the program, the clinical data management and its preliminary information to deliver and to keep up the quality information. I hope it is a great platform to all the research scholars to equip themselves and to excel in clinical research. I wish you all the best for this online mode of conducting the FDP program. I also congratulate Dr. Abdul Rahman, the principal and the professor of Nirmala College of Pharmacy for his untiring work for this task and his valuable guidance to the team. I also appreciate and thank the staff, supporting staff, and all the volunteers for your availability and generous contribution for the success of this program. I also appreciate and thank all the resource persons of the days of this 1 week of online FDP program for your availability and your precious time in sharing the knowledge and your experiences on the clinical data management and its preliminary information to all the delegates. I welcome all the resource persons of the days and all the delegates. I heartily welcome to you all. Utilize this opportunity and equip yourselves. I pray and wish you all the success. Thank you.
Thank you very much, sister, for your wonderful, warm welcome message to all our delegates. Next, we have the man of vision, convener of today's session, Dr. Sheikh Abdul Rahman sir, the professor and principal of Nirmala College of Pharmacy, to give a welcome message to all the delegates. Sir, hearty welcome to you.
So good evening, respected Chief Patron of today's program, Reverend Sister Maria Sundari Garu, Patron, Reverend Sister Nirmala Jyoti Garu, today's resource person, Chitra Madam from SRM University, Organizing Secretary, Dr. Vinay Kumar, Co-Coordinator, Dr. Shreya, and hosting Mrs. and my dear colleagues, faculty, pharmacy professionals. Firstly, happy Teachers' Day to all. So this is the good day. We are starting this ATAL FDP to enhance our knowledge, to upgrade our knowledge in the theme of a novel topic such as novel and contemporary research in clinical research and also in clinical trials. This is the theme of the faculty development program. So before going to the brief discussion of the theme, my heartful and of course sincere and heartful thanks to AICTE for granting the permission to deliver the advanced area topics to our fellow faculty members. So here AICTE ATAL FDP is granted to the colleges. The colleges, those whoever having NAC accreditation A++, and the college which is having NIRF ranking below 100, such colleges only going to get this ATAL FDP grant. I come to know that a total 5,000 applications have been applied to the ATAL FDP, AICTE ATAL FDP, among which they are going to release this ATAL FDP to the 1,000 colleges in 3 phases. In first phase, 250 colleges, 250 to 300 colleges, they were awarded this AICT ATAL FDP. So I congratulate my team members, the pharmacology and the pharmacy practice department, for their hard work, for their sincerity in the process of application. So because of this, today we are in the learning process of the advanced topics. So once again, I congratulate Dr. Vinay Kumar and Dr. Shreya. So because of you people, because of this grant released by AICTE, we are in this, today we are in the online for learning this novel topic. So this, I think ATAL FDP is divided into 2 modes. First one is an online mode, followed by second week offline mode. So you know the timings of anyhow, we have framed the groups, the resource person group and the faculty, fellow faculty group, right? So time to time, our organizing secretary, coordinator of this ATAL, AICT ATAL, sir has forwarded the clear information to all the faculty members and also to the resource person. And I appreciate the efforts made by the coordinator and co-coordinator, because last 10 days we are working on this, and we have conducted the meetings and how best we are going to contribute the novel content to the faculty members. So this is the introductory part of this ATAL FDP, and coming to the today's theme, that is novel contemporary research in clinical research and also clinical trials. So this theme not only supporting or helping to the pharmacology department, and definitely it is going to be beneficial to all the department staff, because the title theme, it looks like a pharmacology and pharmacy practice area, but the way we have planned and the way we have arranged the resource persons, so all the departments, all the contents from various departments, it is going to be delivered in this online and offline mode, so that the faculty members, whenever you are participating in the online mode, I request all the faculty, those who registered or enrolled in this FDP program, so you please take a notebook and pen and write down the information which is going to deliver by the today's resource person and upcoming the topics upcoming days. So that is the most important one, because every key point that is important to the faculty members, right? So these things, so definitely we are going to transform to the junior faculty members and also to the students, so that we have to be very particular and we have to be care about the learning non-topics, right? So that is why I'm telling you to note down the key points delivering by the resource persons, right? And so this is the novel topic here. Resource person is going to present on the advanced software opportunities like CAS, pharmacovigilance, clinical data management, clinical research, right? So in these areas, and also they are going to highlight about the like clinical trials, different phases, phase 1, phase 2, phase 3, phase 4, and post-marketing surveillance. In these areas also they are going to present, and there is a live demo is going to be, live software demo is also going to be present to the faculty members, those who enrolled here. So that I request all the fellow faculty members, so utilize these um, the novel topics going to deliver by the senior most professionals and the faculty, those who expertised in the industry or in our universities or colleges. So we have asked them for their contribution in delivering the lectures in advanced areas. So hope you are going to right understand of today's the inauguration. Thank you, the team members, organizing team members, for giving me the great opportunity on the day of Happy Teachers Day. Teachers Day. So thank you, madam. Thank you, sir.
Thank you very much, sir, for your everlasting support. You have always been with us, behind us, making us to move forward. We are always grateful to you. Now we'll move forward for a message from the coordinator of today's session, Dr. T. Vinay Kumar, sir, professor and head of the Department of Pharmacology and Pharmacy Practice. I request sir to address the delegates and give away the instructions of the online sessions.
Thank you,
welcome
madam.
to you, sir.
Thank you, madam. Thank you. Thank you for your invitation. Yeah, am I audible, madam?
Yes,
Yes,
sir.
audible.
Yes, yeah. So first of all, I would like to convey my best wishes to all the participants, and also Happy Teachers Day to you, all the participants. And I'm honored to be coordinator of this ATAL FDP of 2 weeks ATAL FDP sponsored by AACTE. And dear participants, first of all, I would like to thank you one and all. So those who registered for this FDP and also those who joined for this FDP and making it successful. And as just now principals are just conveyed about the theme and message about the FDP. Just I don't want to waste the time of resource person, today's resource person, Dr. Chitra madam. Just I would like to tell you some few guidelines points about this FDP. Okay, dear all participants, actually this FDP is 2 weeks FDP. This is not like old version. Previously, it was like only 1 week online FDP because of COVID guidelines. It was only through online mode, but now because of decrease in the cases of COVID, ACT decided to keep offline mode also. In this 2 weeks FDP, actually this is called as hybrid mode FDP, faculty development program, right? So in the first week, you are going to learn about the theoretical approaches for the clinical trials and clinical research, both as well as preclinical as well as and also clinical research, the first week. And in the second week, you are going to learn the experiential learning about the topics what you have learned in the first week. Nothing but theoretical approaches. In the second week, you are going to learn about practical approaches, means how to handle the softwares regarding clinical trials, right? The clinical trial design, investigator brochure preparation, and how to handle the SAS and how to handle the clinical data management, and how to do the post-marketing surveillance, and how to design the clinical trial protocol, and how to apply the IND application, how to write the NDA application. All these things you are going to learn by hand in the second week. So dear participants, don't miss the second week sessions also along with these first week sessions. And as per given guidelines by the ATAL FDP, a participant must participate both online as well as offline sessions in order to receive the participant certificate. So at least 80% of attendance you should maintain in order to get the participant certificate from the AACT. So I request all the delegates, don't miss the second week sessions also, which are going to be started from 12th of this month, 12th of September, that is coming Monday. And I would like to tell you one thing that industrial visits we are planning in the second week. So to the nearby industry we are planning. So in that industry, you are going to learn how they are writing the IND applications and how they are releasing the drugs into the market, how they are getting the licenses and approvals, and how they are monitoring after release into the market. That is post surveillance, also post marketing surveillance. Also, we are going to learn in the industrial visit. That industrial visit we have planned on the 12th of this month. That will be in the second. That will be in the afternoon of the 12th, and that's the thing. And up to Friday, we are having next week. I'm talking about next week. Up to Friday, we are having offline sessions, and on the Friday, you will be having one assessment that is through MCQs. And one more thing, participants, please note my point that so there are two MCQs sessions will be there. Multiple choice questions you need to answer. So first MCQ exam will be held at the end of online sessions. That is on the Saturday. On this Saturday, coming Saturday, you will be having first MCQ exam, that is through online mode. And second MCQ exam you will be having on the next week, that is on the Friday. So those who complete these both MCQs, for them only certificate will be generated in the ATAL FDP portal. So please, if you have any doubts, you can contact me. I kept my WhatsApp number and also contact number in the participant group. So because we are already running out of time, so now I would like to hand over the session to the today's host, Mrs. Sasha Madam.
Thank you very much, sir, for your clear-cut instructions. I hope it's all clear for all the delegates. But still, if you have any other doubts, we'll be available for your assistance at any time. You can drop a text to the coordinator or the co-coordinator. So now it's time to introduce the speaker of the day.
Madam, just a minute, madam.
Yes,
Actually, you want a photograph, just to ask all the participants to switch on their video. Madam.
Yes, sir. Yes, sir. We request all the participants to turn on your cameras for a second so that we'll have a photograph. All the participants are requested to turn on your cameras, please.
I request all the participants, please switch on your video. Just will take one photograph. Okay, thank you, one and all. You can now switch off your videos.
Yes, thank you all. Ma'am, very sorry for making you wait for long, as it's inaugural day. We have
no issues. Please
thank you so much, ma'am.
Yeah.
Now I request the chairperson of today's session, Dr. K. Shreya ma'am, to introduce the guest speaker, Dr. V. Chitra ma'am, to all the delegates. Ma'am, over to you.
Thank you, ma'am. Good evening, everyone, and happy Teachers' Day. I'm Dr. Shreya Gosnam, Assistant Professor, Department of Pharmacology and Pharmacy Practice, Nirmala College of Pharmacy. Today, I feel highly honored for this opportunity, and I would like to introduce today's resource person, Professor Dr. V. Chitra ma'am, Dean, SRM University. Madam did B Pharmacy from Tamil Nadu, Dr. MGR Medical University, and she is a recipient of gold medalist and also IDMA GP Naidu Award. And Madam also did M.Pharmacy from same university, Dr. MGR Medical University, and she completed her PhD from SRM University. Madam acted as a chief superintendent of examinations from 2016 to 18 and vice principal for SRM College of Pharmacy from 2013 to 21, and currently acting as a dean for SRM College of Pharmacy. And she also acted as a board of a study member and also head of the Department of Pharmacology and institutional animal ethical committee member and co-coordinator for so many conferences. And Madam has H index more than 15 and has citations nearly 950, and I-10 index is 21, and Madam has published more than 75 papers, and her honored as resource person for various conferences. Madam has received various awards like GP Nair Award, Nageshwar Rao Pantulu Award, and Professor Vain Gopal Endowment Award. Thank you. Ma'am, kindly you can start the session.
So will it be able for me to share the screen?
Yes, ma'am.
Madam, just
Sir.
madam, just I'm disconnecting my screen.
Okay.
Yeah, now you can start, madam.
Those who have questions during the session, you can drop the question in the chat box. I request the chairperson to note down the questions so that we can forward the questions to ma'am at the end.
Sure. And at the end of the session, you will be having like half an hour time to discuss the question and answers.
My screen is visible now.
Yes, ma'am.
Yes. A very good evening to all present here, and a very happy Teachers' Day to all my fellow colleagues. And I feel greatly honored to be a resource person for this ATAL FDP upon the title Novel and Contemporary Research in Clinical Research and Clinical Trials. So it's a 2-week ATAL FDP. I thank the convener, Dr. S.K. Abdul Rahman sir, and the organizing secretary, Dr. T. Vinay Kumar, for sharing me this great opportunity. So I feel honored to be a part of this ATAL FDP. So the title, that is, I am going to share my views upon the phases of clinical trial. So when we come to know about this clinical trials, so what are these clinical trials? So clinical trials are nothing but it is they are studies to test about the new drugs for the purpose of evaluation for their potentiality as well as their beneficial effect in the therapies and the treatments. So this may be for to test new drugs, or else maybe for an already approved drugs. Not only for a drug, it might be for any devices also, or any form of treatment if you want to go for it with the existing drug, with a new form of treatment. For example, the repurposing of drugs in the COVIDs. So these drugs also can be
Madam,
studied.
excuse me, madam. Excuse me,
Yes.
madam. Madam, the slides are not displaying, madam. Only slides are not open, madam. Not open, madam. So can you share whole screen, madam? Then it will open.
It is there in whole screen only.
Okay. Then you can
Now,
select only that window, madam.
now it's okay.
No, ma'am.
But for me, it is showing in a full screen only.
Okay.
Ma'am, once minimize it and reopen it, ma'am.
Yeah, that only I had done it. Now it's okay.
No, ma'am. We could only see the your desktop, ma'am. The files like clinical trial phase. We can only see that. No slides.
Madam, actually, you after opening, madam, you can share the screen. Already you are sharing your screen, madam. But another window you can share where it open. Like new share, madam. Click on the new share, madam. Yeah,
Yes,
now it is open. Now it is visible, madam. Yes, madam. Now it is visible, madam. Continue, madam. You can continue, madam.
Okay. Sorry, sir.
It's okay, madam. No problem.
So yeah. So this is about the clinical trial. So this clinical trial is a planned experiment So this is a planned experiment for the purpose of evaluating therapies or treatments. It might be for a drug, or it might be for a device, or it might be for the treatment of a existing disease with with a new disease with the help of an existing drug. So this many clinical trials, they look at new ways of to detect, diagnose, or else to measure the extent of disease. So some even look at the ways to prevent these diseases from happening. So researchers still they use these human volunteers to test these methods, and the same rules will apply for the volunteers also. In general, so these volunteer controlled trials will be conducted under controlled conditions as possible, so that they provide definitive answers for the predetermined as well as the well-defined questions. Because it's a planned experiment, so we have some predetermined questions as well as the well-defined also. Even after planning of the experiment or the trial, so there will be some questions so which need to be addressed. So this is our drug development frame. So first we need to understand why we need a clinical trial. So the clinical trial, it shows that what works and what does not work. So we cannot, because we have many synthetic molecules in the market, we cannot always say that all the synthetic molecules which are which are trialed to be okay with the preclinical will not be suitable for the clinical trials. So or else it might be in the either way around also. So what works and what does not work in medicine needs to be finalized only with the help of the clinical trials, even though some may prove positive in the preclinical trials. So these are the best way to learn what work in treating diseases like cancer, like end diseases like cancer and then tuberculosis. So all these drugs need some trials. So these thus what it does mean is, or what is the end product is, whether this new treatment is works in people, or if it does, doctors also look at how well it works, and whether this new treatment is a safer one. So all these processes should be developed into a frame. That is the research. From the research, there will be early development. In the research, we have again the three process like discovery phase, candidate profiling phase, and then the clinical phase. And then comes the preclinical, that is a preclinical phase. Then comes the clinical trials like phase 0, 1, phase 2A, phase 2B, phase 3, and phase 4. So these are all the drug development frame. So in this process, so what are all the topics we are going to cover into? So this is about the this drug development frame slide is about the discovery phase, and then the preclinical phase and the clinical trial phase. And in the discovery phase and in the candidate profiling phase, we need to carry out the candidate selection point. We need to select the volunteers for that. And then next is the candidate profiling phase. In that phase, we have to go for the selected for proof of concept. So when we say that, when you include a candidate as a healthy volunteer based upon the study, whether it may be a healthy or a diseased volunteer, so there should be some proof of the concept. So might be of his residence, or might be of the candidate's health status, mental status, or his personal status. The proof of concept is very, very essential. The next thing is about the candidate. The next is the proof of concept is about the disease. What is the necessity of the that is to select these volunteers for this type of disease, and what is the proof of concept? Why do you want to select this group of people, and what are the inclusion exclusion criteria? All those things we need to go for the proof of concept. And then comes the preclinical phase. What we do it with preclinical phase that will undertake the investigational new drug. So when an IND to be the thing, we need the candidate selection part, and then the proof of concept, and then the preclinical phase data. So with that, we have to go for the investigation new drug. Then comes the clinical trials, that is a development decision point and a full development point, and phase 3 is a checkpoint and submission submission decision point. So after that, when the all the phases are all the phase 3 up to 3 phase it is complete, then we'll be going for the NDA, that is the new drug application. So in the clinical trials, so we have the phases like phase 0, phase 1, phase 2, phase 3, and then phase 4. So these are all called as the phases of clinical trials. So the clinical trials, they are usually conducted in phases that build on one another. So each phase is designed to answer certain questions of that area criteria. So knowing these phases is very important because it can give some idea about how much the treatment, the drug for the which is available for the treatment is being studied among the population. So there are even benefits and even risk also will be there when you are passing over from one phase to another phase in a clinical trial. Although these clinical trials are devices as well as for other diseases and treatments, they are used in the examples of clinical trial phases. So this preclinical to clinical studies. So this is the transition type. So this preclinical, we know that this is about we pharmacologists do is upon the preclinical trials. So these clinical trials can be done only after finding out the data of the preclinical. So only then the IND can be given. That is, the treatment is likely to be safe or whether it will work with the people or not. So these preclinical studies, they are also called as laboratory studies, which include the cell culture as well as the animal studies. So this will give a lot of useful information, but not that whatever is needed, because it is like a raw data. And this because the humans as well as the rodents will vary in many ways, though all belong to the mammalian category. So their variations will be there. So the treatment that works, that's what I was saying you in the beginning. The treatment which work out in a rodent may not be beneficial in the human being also. So there could be some untoward side effects or any other problems which could not be experienced or which could not be revealed in using preclinical studies will be really revealed in a clinical studies. So this is the transition. So on completion of this preclinical, the number of lead compounds, so might be of a synthetic origin, semi-synthetic, or else from the plant or origin. So many fewer useful candidate of drugs will be there. So these useful candidate drugs can be sometimes may proceed to an advanced treatment or else to a clinical development, so which involves testing in humans. When we see some extraordinary results of the synthetic molecules or a semi-synthetic molecule in a preclinical trial, so that can be involved testing in the humans. So these data on the preclinical studies must be submitted to the regulatory authorities for the registration of that particular compound. Supposing if it is a curcumin, when we say that curcumin is a very good compound, investigational drug, when it is found to be effective in a preclinical study, so then it can be registered as a compound as a investigational drug, IND. It can be given a investigational drug, and permission from the regulatory authorities will be sought only after confirming the whatever the data which has been obtained from the preclinical studies, and after obtaining the undertaking studies in the human. So this is the transition. So this preclinical data, whichever is required to be submitted to the regulatory authorities prior to the human study. So what and all we need regarding the compound, whatever we claim that as an investigational drug. So what data we require is the chemistry and the physicochemical properties, chemical properties and the physicochemical properties of the drug, and the formulation data. What is the type of formulation we are going to give? Might be what is a liquid or a oral formulation or else a injectable, whatever it is, the formulation data, as well as the components which are present, and the details of the method of manufacture. So how the compound has been synthesized, whether it is a micro oven synthesis or it is a green chemistry or whatever it is. So what is the method of manufacture, and whether it is a labile compound or a thermostable compound, and its analytical specifications to confirm that this compound, whatever you are claiming to be, is that. So we need the analytical specifications and a general pharmacodynamic profile, and then the specific pharmacological efficacy, what we have claimed upon the laboratory animals, and the kinetics, what are the kinetic study, that is the absorption distribution pattern, and the drug metabolism, how the drug is going to get metabolized, and the toxicological studies. So how far the drug is safe whenever it is given to a rodent population, whether there are any lethal effects or harmful effects are seen upon any of the major organs. So this is the data which we required to be submitted to the regulatory authorities before you design a human studies. So once you get the authority, that is regulatory authorities, approve these data, and they'll be giving permission to proceed for the human studies after getting a specific undertaking. So in 2006, so this FDA has issued a guidance document regarding the exploratory IND studies. So this exploratory IND trials, that included the pharmacodynamics and pharmacokinetic testing. So this phase 0 is a recent designation for the exploratory and first in the human trials. It is known as human microdosing. So given in micro amounts, and then we have to see for it. And it is designed to speed up the development of the promising drugs. So even though this phase 0 studies are done in humans, this type of study is not like the other phases of the clinical trials. So the purpose of the study is to speed up and to streamline the approval process. So the phase 0 studies, always they use a few small doses of that is only is called as micro dosing. So it use only a few small doses of a new drug in a few people. So dosing is also micro. The people, that is the population selected, is also a few people only. They might test whether the drug reaches the particular targeted area and how the drug acts in the body. That is about the pharmacodynamics and the pharmacokinetic testing. So unless there is that is the other phases of the clinical trials, there is almost no chance the people in the phase 0 trials will benefit. So the benefit will be for other few people in the future, because we may not know because the drug doses are very low. So the risk also will be very low, because it is a micro dosing. It is given in a few doses. So we may not expect, that is the testing population may not experience much of the serious adverse effects because of the low doses. But then to start with, you have to, is been given only with fewer to 15 people only. We are given to only 15 people to confirm whether the drug behaves in human subject as it was expected from the preclinical. Just like a trial, like a pilot study, we are giving it. And then next is that is a phase 0 study conducted on a limited number of subjects, that is 10 to 15, involve a very small dose, have a limited dosing duration, only 7 days, having no therapeutic intent, just to see whether the medication or whatever the newer drug is being not a drug, it is a lead compound, whether that compound is being safer to the population or not. So conducted before the traditional phase 1, that is, it is even a pilot study for the phase 1 studies of dose escalation, safety and tolerance. So that is the reason why it is given only to a 15 group of people. Purpose is to gather the preliminary data as on the agents, pharmacokinetics and the pharmacodynamic profile. So this takes about 9 to 18 months to complete and involves about 20 to 80 normal healthy volunteers. The age group is from 18 to 45 years. They are conducted to determine the appropriate dose range with regard to safety and toxicity. And they are used to document the human drug metabolism and mechanism of action. They may also be conducted in severely ill patients. So these phase 1 clinical trials, it is usually the first that involve people with the, that is before, the phase 0 was not there before. So it was the first phase which was used with people. And these phase 1 studies are done to find the highest dose of the new treatment that can be given safely without causing severe side effects. Although the treatment has been tested, then too we need to show about the disease. So what all the exclusion is children. Children are the exclusion criteria, and women of childbearing age, and unless the nature of the IND necessitates, their inclusion will be there. That is example is the oral contraceptive study and in the elderly. So they are not included for this. So the key points for this phase 1 trials are they get a very low dose of the treatment and are watched very closely. And if there are any minor side effects, the next few participants get a next higher dose. So when they are checking with the first lower dose, when there are no side effects, then the next group of people will be going for the next higher dose. So this process continues until the physician find that this dose is mostly likely to work without having a maximal side effect. That is the acceptable level of side effects with the maximum level of efficacy. So till that the doses will be higher, and then it will be given to the next group of population. So this safety is the main concern here. So the team should keep a close eye upon the people and watch over for any of the side effects. And because of the small numbers, again there may be rarer side effects and may be seen in the later stages of the phases. Actually, in the phase 1 trial, we don't have any placebos. That is, inactive treatments are not there. We are directly going for the thing about the drugs again. So again, this is going to be for a small number of people only. They can be with the severely ill patients also. So these endpoints of the phase 1 trials include, that is whether the drug to be given in a single dose or it can be given in a multiple dose. This is the results or the outcomes of the phase 1 trial. So the determination of the single or multiple doses and the dose range and what is the route of administration can be given, and the pharmacokinetic data, dynamic data, and what is the maximum tolerated dose, because as it is given to a smaller group of people with smaller doses, when only that smaller group of people, when they experience any adverse effect, then the doses will be stopped from moving to the next generation, that is to the next group of people. If not, then again it will be given to the next group of people with the next higher dose. So from that we can just measure the maximum tolerated dose as we do for the rodents also, and the other parameters whatever it is kept necessary for the ending of the trials. So this phase 2 trials, so this these trials, they start with the low doses, and it is the fractionation of the clean or no effect dose when it is observed in the toxicology studies. So the low dose, how to start with the low doses? It might be with the no effect dose. It is called as the no effect dose, which is observed in the toxicology studies. When we are giving, like in the toxicology studies of 10, 20, 30, like that, when we go for a progression up to 100 milligrams, say for example, might be in the 10 and in the 20 we see no effect. So then it should be the starting dose for this type of trials. So here the unwritten rule is dose should be 125 or 100th of the no effect dose in mg or kg. Supposingly if it is a 10 mg, then it should be 100th or 125th of the dose. So the patient should be monitored for the adverse drug reaction. So this phase 2 clinical trials. So this begins after the satisfactory or the preliminary evidence of safety data being obtained. So this involves the supervised administration of the drug. So this should be done under supervision, because we are going for the next phase. There may be the occurrence of any of the adverse effects or untoward effects may be there. So it should be involving a supervised administration of the drug. So that is this involves for treatment or prophylaxis against a disorder for which the drug is being intended. So it is to about 100 to 300 patients. This phase 2 is for 100 to 300 patients, and usually it is a randomized study comparing the new drug with the prototype drug for the intended disorder. New drug, whatever the drug, supposingly if it is an analgesic drug or an anti-inflammatory, so you are just going to compare this new drug moiety with the prototype, might be an aspirin, or if it is with paracetamol, or if it is with diclofenac. The study should be compared with that of the prototype drug for the intended disorder, whatever the disorder you intend to screen upon. So it provides a first opportunity to observe the effect of long-term administration of the drug to humans. So in this first, phase 2 only, we can observe the long-term administration, that is the long-term effects of the drug when it is being given to the humans for a long time. So it is for a group of some 25 to 100 patients can be given. So usually that is everybody will be getting the same dose, and these may be done in these phase 2 trials can be done in a communities and hospitals, or else maybe in the any centers. For example, if it is cancer, it can be done in a major cancer center. Here also we do not use any placebos. The purpose of this phase 2 is to determine an optimal dose, because in the phase 1 itself we have just noted down which dose is not toxic, and because we are just done a pilot study with small group of population, and we have calculated the maximum tolerated dose. The purpose of this phase 2 is to determine an optimal dose and the response range for the new drug. So what will be might be up to 20 milligram, the drug is not showing any of the toxic effect, or even the beneficial effect is not being shown. Then the toxic effect seems to be that is expressed only after 40 or 50 milligram. So the response range will be measured. This is the phase 2. And to verify its efficacy for the intended disorder. Suppose if it is for a painkiller, or if it is for a cancer pain, so how the efficacy is there for the drug for the intended disorder. So the participants will be also monitored for the adverse effects, because the population here is large, more than 100 patients will be there. Hence, there will be greater chances of detecting additional adverse effects other than the normal adverse effects, because the population genomics will be different. So the variations will be different, genetic variations may be different. So the even the adverse effects also will be, we can be observing the additional adverse effects also. So the crucial phase is the data is used to determine whether to proceed with the exemption. So this is a very important step in the phase 2. That is, it is very crucial to determine whether this study is okay. Up to this, we can stop, or else it is safe to proceed for the next phase. So that is the reason why it is called as a crucial phase, because it is patients is also large, and less common side effects if it is seen, then it is safer to proceed for the next phase. Otherwise, within the phase 2 itself, the trials can be stopped, and even the research scientists should look about what are all the points we have to take it, and what are all the adverse effects it is seen. So how to go for that, how to go for decreasing or nullifying those adverse effects. So this types, again, here we have 2A and 2B. Again, we have seen about the proof of concept studies and definite dose finding studies. And in the type of clinical trials conducted are the pilot trial, that is the test drug in, that is a pilot trial of testing the drug in patients. Maybe either it might be an open label, or a single or a double blind manner. So either of the, that is the, only the physician will be knowing, or else a physician and the investigator will be knowing. It might be a single or a double blind manner. So it might be a pilot trial of the test drug in normal volunteers. So first phase is in patients, second will be in the normal volunteers. So if it is challenged to determine the efficacy of the drug. So example, the citric acid may be used in the normal volunteers also, it may be used to produce cough. So this is a dose response trial in which patients will identify the range of active doses. The variations in the dose level or dosing schedule can be monitored, and the duration of the dosing required to achieve a sufficient magnitude of efficacy. Supposingly, if the drug effect is going to decline in the plasma or else in the blood within the first 2 hours or within the first 3 hours, or the drug is effective for more than 6 to 8 hours. So what is the frequency of the dosing we required for the drug or else for the ailment to be achieved a sufficient efficacy? And then the dose interval trial, which evaluates the time between the doses. So every 6 hours, for example, if we take when a heavy fever, dose 650 at every 6 hour intervals. In a normal fever or in a normal headache, we usually take only once a day or twice a day. So what is the time between the doses we need to understand? So the phase 2B, so this is a type of clinical trial conducted is to determine the optimal dose. So in that we are determining the doses and at which we have a maximum efficacy. In this we determine the optimal dose at which the drug shows biological activity with minimum side effects. That is a dose range finding test. So this is a optimal dose at which the drug shows biological activity with minimal side effects. The biological activity also will be there. The side effects will be minimal. So this is also called as pivotal trials. And this is for the test group. That is how do you classify this? That is the like a schedule. Phase 1, it is a test group is 20 to 80 healthy volunteers. And for the purpose is to establish the basic safety and the blood levels achieved with the different doses of the drug. So duration 1.5 years. Phase 2A and 2B, it is 100 to 300 people who have the disorder. For example, if it is a diabetes or if it is cancer or hypercholesteremia, hyperlipidemia, arthritis, whatever the disorder may be. So it includes 100 to 300 people who have the disorder being studied, and to establish the drug's effectiveness and the dosage range, to determine the drug kinetics and to identify the side effects, and the duration is 1 to 3 years. Next comes the phase 3 trials. So the government has permitted the pharmaceutical companies to conduct concurrent trials in India as well as in overseas, as there may be the variations will be there. So the treatments that have shown to work in phase 2 clinical trials must succeed in one more phase. So that is the phase 3 trials. So these phase 3 trials will compare the safety and effectiveness of the new treatment against the current standard treatment. Okay, the new treatment, how far this new treatment is effective against the standard treatment which is available now. So which it is usually a randomized. The participants are often picked at random. That is called as it is a randomized studies to get either the standard treatment or the new treatment. So whenever it's possible, the doctor nor the patient knows which of the treatment of the patient they are getting. That is it might be a single blind or it might be a double blind. So randomization and blinding studies are carried out in the phase 3 trials. Phase 3, it is a full-scale evaluation. It involves the most extensive testing to fully assess the safety and the efficacy and the drug dosage in a large group. So the phase 3 trial is most rigorous and it is an extensive type of investigation. The objective is to compare the efficacy of the new treatment with the standard regimen. Design is a randomized pick from the patients will be randomly picked and the subjects will be several hundreds to thousands of patients with deceased volunteers, and the sample size depends upon the various factors which need to be included and which need to be excluded. The duration of the study is 5 years. So if the FDA feels that it is more needed to show that the new treatment benefits outweighs its risk, so it may ask for more information and it has all the right to accept or to reject the studies. So this expanded clinical trials, that is a multicentric trials. Multicentric trials are used, that is where the larger number of patients can be used. which can be done in various parts of the country or in various countries also. So to compare with the previously established therapy or else the placebo therapy, so both are going to be nullified or whether the risk is being outweighed, as well as to demonstrate the effectiveness and the safety. The next is to determine the benefit and risk ratio. These trials are started after the NDA submission and to determine the additional safety data and to determine the therapeutic benefits and to determine the additional indications, maybe of the any other additional indications also. Expanded controlled clinical trials. So the phase 4, so this phase 4 is a very important phase, that is up to phase 3. We are doing it in a closed population as well as in a closed, might be in a closed circuit. But this phase 4 is a post-marketing surveillance. Most of the drugs has been withdrawn in the market after this phase 4 studies. After the drug is released for general use, that is a post-marketing silence, to be completed within 2 years. So rosiglitazone and most of the drugs like cimetidine and these drugs has been withdrawn from the market because of this post-marketing surveillance, because we need some data for the, because see even the physicians may also have this coded drugs, so which will be given for trials, that is a post-marketing surveillance. Even the drug will be released in the after even the phase 4 studies is over, when the drug is being given to the market and when it is being consumed by a variegated population. So those population, some of the population or some of the genetic factors in the population will not have it in an acceptable rate, so that the medications will not be tolerated by all. So it determines the real clinical status. This is what we can say, that it determines the real clinical status and it determines the nature and the frequency of the ADR in diverse clinical settings. So for different group of populations, how this drug is working upon. So if it is an anti-diabetic drug, so they would have concentrated much upon the anti-diabetic drug and least concentration over the cardiovascular effects. But this beneficial effects is overweighing the risk. So the risk is the cardiovascular toxicity. This rosiglitazone like drug, so when it is giving this when it is precipitating the cardiovascular risk, so that is the reason for those drugs to be withdrawn from the market. So like this is the this is the real clinical status. But as of this thing, even for your pyroxicum, so this pyroxicum is not safer for the children. So for the children, it gives that is, it precipitates hepatotoxicity. It is not recommended for the even your nimesulide. Nimesulide like drugs are not recommended mostly because of the, but it has an excellent analgesic property. But this is not recommended for the patients with liver toxicity or the children. It should not be given to the children. So these are all the that is the risk which has been that is what is explored by because when after the that is the drugs has been circulated in the market. This is called as a post-marketing surveillance. So it determined the nature and the frequency of the adverse drug reactions, as well as to recognize the unanticipated additional benefits. Might be if it is an additional benefit also might be there, which may result in additional or secondary uses for drug. So metformin, when it was introduced, it is only an anti-diabetic drug. But when it is being given to many populations, one group of population says that it is good for a PCOS also. One of the population now saying that it is good for cancer even. So like all-in-one drug, this metformin is giving some good treatment regimen for most of the hormonal related problems. So these are all the secondary uses of the drug. So this is an unanticipated as well as an additional benefit. So this is about the phase 4 clinical trial. And this is how the phase 4 is being distributed. This is given to the normal population and we are giving it as a. So now that is your periodic safety update reports. So this is your last commitment. That is your periodic safety update reports. PSUR is very essential. They have to be submitted every 6 months for the first 2 years after the approval is granted to market the drug. So when a drug we have applied, IND status is being converted to NDA. After this NDA, the regulatory authority has given the moiety as a as a designated drug. So now it is being assigned to market the drug, and thereafter, for subsequent 2 years, the periodic safety update reports has to be submitted annually. That is 2 times a year, which can be further extended if the licensing authority feels so in interest of public health. Supposing if the drug is to be dealt with in any anti-diabetic or with an anti-cancer, it needs at least a 5-year report. So new studies specifically planned or conducted to examine the safety issue should be described in the update report. So whatever it is, might be if it is a drug and after some 2 years or 3 years, it was found that it is going to be benefited for some other population also. Then if there is specifically planned for a new study, it should be conducted to examine the safety issue, which should be described in the PSUR, that is in the periodical safety update report. For all non-serious spontaneous adverse effects noted in the clinical practice as well as in the post-marketing clinical studies, it will be submitted through a PSUR covering only the period of report. That is called as the interval data. If there is non-serious or any spontaneous, might be even after taking it, there may be a sharp headache. Only that is, it is a non-serious, but when it is a spontaneous adverse effect, it should be also noted it down and it should be submitted to the that is to the authorities covering only the period of report. That is during this period, that is the drug is having this data. This is called as a periodic safety update reports. So this is all about the phases of the clinical trials, and our ultimate goal is to safeguard the public health or the population against the, that is before a drug moiety enters into the market. Thank you.
Thank you very much, ma'am. That was a very clear session for all of us. Now I'll forward to the chairperson, Dr. Shreya ma'am, for the questions. Ma'am, please go ahead with the questions.
Yes, ma'am. Ma'am, there are a couple of questions from audience. So what are the ethical concerns when it comes to recruiting the subjects in phase 4 or microdosing studies, as it is not beneficial?
So this microdosing study is always beneficial. It may not benefit the community, but it will benefit the that is the investigator who conducts the trial. So before a series of events, for example, you can take the thalidomide tragedy. So this thalidomide, when it is not being it is not being tested well in the pregnant population. So then this tragedy has happened. But after this has happened, we cannot control anything. That is the reason why we are giving it as microdosing. Whether it could affect only a small group of population, it may not be beneficial therapeutically, but for an ethical concern, it is beneficial. It should not affect the large population or the huge population as a whole once it goes to the phase 2 trials. That is the reason.
Okay, ma'am. Next question is, who are normal volunteers in 2-year studies? Is it patients with clinical condition or healthy volunteers?
Healthy volunteers. Normal is always healthy volunteers. There we have two sort of patients. Sorry, one will be the two group of set of people. One will be the healthy volunteers. Next will be the diseased population. Both will be there.
Okay, ma'am. Thank you. Is anyone from the audience having any questions still? So Vinay sir.
Yes, madam. Madam, thank you for your excellent session, madam.
Yes.
For this pages of clinical trials. That was a very informative session given by today's resource person, Dr. Chitra, madam. And one more session is there, madam. So
Yes,
will you continue, madam, or will take rest for 5 minutes, madam?
That is up to you. I don't have any concern. If I want, if it can be, I'll be okay, sir.
Okay. Madam, actually, one more doubt from the audience side, Madam Prashant Kumar. Ma'am, in which phase drug food interactions can be evaluated?
Drug food interactions cannot be evaluated might be in the phase 3. That too with the controlled conditions only we can go for it. Only these drug food interactions will be more beneficial in the phase 4 trials. That is in the phase 4, which is not under our control, but in the phase 3, it is necessary to go for to find out the drug food, normal food content, whatever the volunteers take. So those drugs, how the food is getting interacted with this phase drugs. So that will be there in the phase 3 and in the phase 2, because we do not go for any drug interactions. We don't go for it, but drug food interactions can be there in the phase 3. And more what is exponentially, if we need, we have to see in the phase 4 trials only.
Okay.
Thank you, ma'am.
Thank you.
Thank you, sir. Ma'am, it's 8:20. Shall we take a break of 5 minutes and then start by
Yes.
8:25?
Yes, yes.
Yes, ma'am. Thank you so much, ma'am. All the
Thank you.
delegates, you can take a short break and be back in 5 minutes. Ma'am will be ready for the
Yes.
second topic, ethical principles and clinical research.
Okay, thank you.
Dear participants, so just I'm sharing the list of sessions that we are going to listen in this first week online sessions. So please go through that slide share. Just now I'm playing the slide share within
Hello.
this 2 minutes.
Hello. Yeah.
Hello, madam. Asha, madam. Shall we start the session, madam? Second session.
Yes, sir. I think ma'am is not yet here, sir. So I'm waiting for ma'am.
Okay. We'll wait, ma'am. We'll wait for madam.
Yes, sir.
So, meanwhile, madam, can we? Yeah,
Sir,
yeah,
ma'am
madam,
is-
ma'am is here.
I
Yeah.
am here, Vinay. We can start.
Yes,
Yes,
ma'am.
yes,
Welcome
madam,
back
yes,
to you after a very short break, ma'am. Thank you very much for your patience and energy on the session. So let's, dear delegates, we'll move to the second session of the day, ethical principles in clinical research. Ma'am, over to you, ma'am.
Yes.
Yes, ma'am, we can see your screen.
Yes, yes, ma'am. So, very good evening to the participants, and we shall start with the second session of this FDP entitled Ethics in Clinical Research. So, the last session we are dealing with about the phases of the clinical trials. So now it is time for us to know what are all the ethics we should follow when we do a clinical research. So this focuses largely on the identifying and implementing the acceptable conditions for the exposure of the individuals towards the risk or towards the burdens for the benefit of the society. So they are doing it, as we say, and as we acknowledge the that is the laboratory animals who are losing their life for a noble cause. The same applies to here. Here also we should we are liable to follow certain ethics, because those population are being exposed to the risk and burdens knowingly for the benefit of the society at large. So these ethical guidelines for this clinical research were formulated only after the inhumane behavior, because when everything is going on ethically, there is no need for any formulations or any guidelines to get followed up. When these regulations are going to be inhumane or the behavior is going to be unethical, then there came a time where they have to formulate certain ethics in the clinical research. That is when it involves the participants, when they are using it in a research environment. So this word ethics is derived from the Greek word ethikos. So this ethos means a custom or a character. So this ethics is nothing but it is a systematic study. It is a systematic study of values, maybe of whatever it might be a human value, or it may be of a moral value, or it might be of any of the ethics. It might be a traffic ethics, or it might be what all the anywhere where it has to be followed very systematically. So this ethics is a systematic study of the values which has to decide what is right and what is wrong. So these are the moral principle which governs the human character and conduct. So that when it comes to this medical ethics, this is the ethical guidelines which governs the healthcare system. Might be a medical practice, that because there are some ethics with the medical practice also, some ethics which concerns with the medical research also, and there are some moral values and judgments also when they apply to medicine. So there are most of the medicine, this thing, whenever it is being given, there is a ethics that it should be counseled before giving a medicine to the that is to the patient or to the volunteer, whomever it might be. It should be said very clearly that how to take, when it has to be taken, and what is the frequency of dosing, or before food, after food. These are all like ethics what has to be followed when it is to be given to a medicine. And when it is going to be a research, when the research is going to be conducted, then we should know what are all the ethics what we should follow, what and all we have to take care of, what and all we can be we should not neglect certain things, what are all the eligible this is affordabilities what which has to be given to the that is to the volunteers. So these things are what are all the ethics. So this is what has been so these ethical guidelines they were formulated one, that's what I told you, only after the inhumane behavior these ethical guidelines were followed. These were carried out in the pre-World War II era. So most of the research experiments were carried out on own self or else their own patients. The doctors used to do it on their own patients. So this World War II, it leads to take more interest in the science and research, because many people were dying because of the lack of antibiotics, because of the infections. Many people were dying. So this is the reason why there came a ethical guidelines. So the Hippocrates was the first physician who defined the ethical principles of research in humans. So before that, there was no ethical principles were there. So the experiments, that is, these principles are still valid today. That is the autonomy. Autonomy is their self. So to respect the autonomy of the participant or of their representative, whether the autonomy is, that is, if I am the participant, then I should be given, my decision should be given some value. If I am willing to participate, if I am not willing to participate, that decision to be respected. Or if I am not able to speak or express myself, my representative should be given the due value as I was given. And the beneficence is to act always in the best interest of the participant. So the participant should be benefited out of the study, whatever it is. So those beneficial ways also to be explained. And then the non-maleficence, so to do as little harm as possible to the participant. So we know that there will be less effects will be there, or less harm will be there to the participant. So that also should be explained. These principles to be explained. And justice is to act fairly to all. Because I have formulated a medicine, I have applied for a synthetic molecule, I should not claim that my drug is good, and I should not act unfair with the participant. So that if there are any adverse effects are there, I should be able to, in a position to report that, and I should be in a position to rectify that. I should be in a position to take care of the participant after it has been given the whatever the drug of experience. So what is the this thing, medico-ethical? What is this? During this World War II, that is the Nazi region, that is the Nazist regime in Germany, the human medical experimentation was done in large number of people in its concentration camps, where their group is going to be in a higher number. So though in those groups, the medical experimentation was done. In particular, that is, the experiments were done on prisoners. They earned him the nicknames of the Angel of Death and Dr. Death. So the experiments were done on prisoners. So that is why he is called as the Angel of Death or Dr. Death, because there is no a preliminary data or no previous experience that how the drug is going to act upon the particular individual. So between 1913 and 1945, this Japanese medical personnel, they participated in the torture killing of as many as 10,000 Chinese and Korean prisoners during the Second Sino-Japanese War. So this is the condition in the experimentation using human volunteers. And there comes the end to that show. And after that, it was framed by Hippocrates about the ethical guidelines, what has to be followed in a clinical research. So these, the death was the end point in most of the experiments. That is why it is called, he is called, that is, the investigator is called as the Angel of Death, or else the Dr. Death. So this is the doctor also did antemortem dissection to study the changes. So that much, what is the unethical way was being practiced before these guidelines were formulated. And there comes a point where the ethical guidelines are to be formulated, and it has to be followed before a drug to be given to certain group of population. And this is the starting of this ethical research. That is again, the same point what I have told is being repeated in this. During the Second World War, the Japanese tried out various biological weapons on Chinese subjects, and these Japanese surgeons also performed vivisection and other medical experiments to torture the American prisoners in several islands of the Pacific. So the following are some of the experiments which has been conducted by Nazis, which led to the Nuremberg Code. So these are all the experiments, that is, experiments on twins, freezing experiment, malarial experiments, mustard gas experiments, sulfonamide experiments, seawater experiments, sterilization experiments, experiments with poison, incendiary bomb experiments, high altitude experiments. All these experiments were tried using the prisoners. So then came that is the discovery of these experiments stunned the whole world. So then it led to the Nuremberg Code in Germany to prevent the recurrence of such episodes. So it should not recur again. So it was the first international code for ethics in the clinical research, laying down the guidelines for research on human volunteers, that is upon not volunteers, human subjects. So it laid down 10 clear principles to be followed by the research and made voluntary consent essential. Before that, there was no practice of getting a consent form. But after this Nuremberg Code was being formulated, so it is essential that for these experiments, there should be a consent to be get from the, that is from the volunteers, and the allowed subjects to be, they have to allow subjects to withdraw from the experiments even after that is in between the experiments also they can withdraw from the trials or else from the study if they are not willing to do so. So the banned experiment should not be repeated and the volunteers should be allowed to withdraw in between. So these are all the that is the ethics they had followed. That is after framing this Nuremberg Code. So this Nuremberg Code, this verdict is delivered in August 19. This was not honored by many of the researchers, and there continued to be some exploitations of the humans in research. And then that is Dr. Leo Alexander and Andrew Canby. So they drafted again a 10-point memorandum. So this was called as the permissible medical experiments. What are all the medical experiments can be permitted? Even after that, there was some experiments which has been done under that is unethically. And after this again, that is a World Medical Association to develop a fresh set of guidelines to safeguard the rights and the well-being of the participants in the that is in the study. So it was received 5 times, and the latest version was published in 2000 at the 52nd World Medical Association in Scotland. So the Declaration of Geneva, which was adopted by the World Medical Association in Geneva in 1948 and amended, that was, I told you, about 5 times, about 10 times it was then again revised, and finally it came to about in 2000. There was a verdict, and again it is declared by the physician's dedication to the humanitarian goals of medicine. It was especially important in the view of medical crimes which has been just committed in the Nazi Germany. So the Declaration of Geneva was intended as a revision of the oath of Hippocrates to a formulation of his moral truths. And after this thalidomide tragedy, so this Harris Amendment was passed. It is responsible for the current investigational new drug application regulation. That is a voluntary consent of all subjects in non-therapeutic and therapeutic research. It became mandatory. So before that, the consent was not mandatory, but after that, the consent became mandatory. The drugs were required to have proven efficacy and safety. Without any preclinical data, the drug should not be submitted. That is, it should not be submitted for any approval. And FDA approval was required for marketing a drug. So this should be approved, and all NDA should be reviewed by FDA. So all the new drug applications should be reviewed by the Food and Drug Administration. And the Declaration of the Helsinki, it was first published by in the World Medical Association 1964. At the heart of the declaration is a statement that the well-being of the human subject should take precedence over the interest of the science and society. So it should be taken in the interest of the science and society. Only if that is going to be the case, then it has to be tested in the participants, maybe in the healthy or else in the non-healthy volunteers. It also gives special attention to the importance of written informed consent. So before that, there was no, as I explained you earlier, there was no informed consent. So after that, there's declaration of Helsinki, there was a consent to be taken from the volunteer or from the participant whomsoever is going to get participated in the study. So the next is the study of untreated syphilis trial. So this is a African American men with the latest with the latent syphilis is being enrolled. So this trial aim was to study the disease course of syphilis, because penicillin is recognized as a recommended treatment for syphilis in 1947. The participant did not receive the available medical treatment, because the malpractice in the syphilis study in the US was possibly because all the participants will not be getting this penicillin, because it was then recently developed and it was not available to all the patients who have this syphilis. So this malpractice in the syphilis study was possibly the next eye opener, which ushered the Belmont report in 1979 and laid the foundation for regulations regarding the ethics and human subjects for the research in the US. So these are all the important declarations. That is a Nuremberg Code, and then the Declaration of Helsinki, and the National Research Act, the Common Rule, and the Belmonte Report. And in 1982, WHO and Council for the CIOMS issued the international guidelines for the biomedical research. And next is the International Conference on Harmonization Guidelines, and the Indian Council for Medical Research had laid down the ethical guidelines for biomedical research on human subjects. So these are all the guidelines. So first is here ICMR ethical guidelines for research to be, that is the Indian Council for Medical Research. This is the research to be conducted ethically. We need to follow 12 general principles laid down by the ICMR. In order to follow these, we should be aware about the informed consent process and the vulnerable population. Vulnerable population is who are the population which needed to be treated for this type of ailment. And the therapeutic misconception. So what is the therapy to be given, but it's misconceived and the treatment is given in a wrong way. And the post-trial access and the structure and role of ethics committee. So these are all the process we need to be aware before we move on to the ICMR guidelines. The principles of essentiality. So here the research is carried out should be essential for the advancement of the knowledge and for the benefit of all members of the human species and all the others in the aspect of healthcare, and also for the ecological and environmental well-being of the planet. So the principles of essentiality is needed. That is why this study is going to be carried out, whether it is going to be necessary for the advancement of knowledge, or else it is for the benefit of the human species, or whether it is for the ecological or the environmental well-being. It should support any of these facts. Otherwise, it is not mandatory to proceed for this essentiality. That is, it is not going to satisfy the first principle. So second principle is the principles of voluntariness and the informed consent and community agreement. This voluntariness, this research subjects are fully apprised of the research and the impact and the risk of such research on the subjects and the others. So this is called as voluntariness. So if I am a volunteer, I should be explained about my role and responsibility towards this study or else towards this research, that how I am going to be useful and what is going to be my role and what will be the rewards I am getting out of this thing. If I'm participating in an event, I'll be asking about when I have to report, what should I do, what is the dress code I should wear, and what will be the benefit, whether I'll be getting a certificate out of it. as a student, everybody will be having this thing. Where if it is going to be a volunteer or to be a subject in a particular study, then that voluntariness will come only if the that is the whole process is being explained very clearly. So it should be given full appraised about the research. So the research participant should be aware about the nature of the research, as well as he or she should make a independent choice without the influence of any doctor or any investigator. If I am the investigator, I should not go and say that no, the study will be benefiting you, you will be getting these benefits, that benefits, and we should not talk about only the benefit. I should say him also about the nature of the risk which is being involved, not to threaten him, but to make him aware, or else to make him sensitized about the treatment procedure of the schedule. So when the research treats any community or any group of persons as a research participant, these principles and the informed consent should apply to the community as a whole. Not only you cannot voluntarily, you cannot pick up one participant and you cannot get it from them, to be got from every individual, and to each individual member who is the participant. So the research subjects retain the right to abstain from further participation. If I'm not very happy about the way the research is going on, or if I'm feeling somewhat uncomfort, or my mental status is not permitting me to proceed with this study further, then I'm having the right to retain the that is, I have the right to abstain from the further participation. So for that, they'll be having some added, like reserve participants they'll be having. When I am going out, there will be some reserved participants who will be participating in the study. So when the human subject is incapable of giving any consent, or it is considered essential that research or experimentation be conducted on such a person incompetent to give consent, the principle of voluntariness and informed consent shall continue to apply, and such consent and voluntariness shall be obtained and exercised on behalf of such research by someone who is empowered under a duty to act on their behalf. If I am not in a position to give a willingness or to for the consent or to give a consent, so any representative of my side, maybe of my relative or my friend, whomever it is, whom I consider that as my nominative or on my behalf, he or she can be she can give the consent on behalf of me. So principles of non-exploitation. So the research participation should be remunerated. Out of the remuneration, so when we participate in a study, the research participant should be said that if you are a daily basis, whether it is going to be a 10 days or else it is going to be a 6-month process, then they have to be given some remuneration on a weekly basis, or else on a daily basis they have to be remunerated for their involvement, or else they can be given a that is some slot of money, so that for this 6-month study you'll be paid 10,000, so that along with the food whatever we give, everything will be in a controlled atmosphere, the controlled food condition, everything we have to, it has to be explained to them. So the participant should be made aware of all the risk involved. So he has to be irrespective of the social and economic condition or education levels attained. So we should not say that the candidate or else a participant is an illiterate, or they are very weak in their economic status. So we should not exploit the condition of the volunteer, whomever it is. We should not exploit their social nature, or maybe they may be socially deprived or economically deprived, or they may be an illiterate, education levels are very low. So that is the investigator or the physician should not utilize or exploit his or her condition. And every research protocol should in provisions of compensation of the human participant, either through insurance cover or any other appropriate means to cover all foreseeable and hidden risk. So might be there should be some compensation should be given to the human participants. That is, there should be surely an insurance cover should be there, or any other appropriate means to cover all foreseeable and hidden risk, because we cannot see everything. It may happen or it may not happen also. Like a insurance, what we take, that after 20 years, we are going to get this much amount of money. Only God will be knowing after 20 years, the particular individual is going to be there or not. Okay, but what to say, we are just keeping a what is a positive mindset. We are just investing in the insurance coverages for getting all the benefits after 20 years. So like this here also, to cover all the foreseeable and hidden risk, the participant should have some insurance coverage. And the principles of that is here, privacy and confidentiality. So the identity and the records of the human subjects of the research or the experiment should be kept confidential. So might be the data whichever is acquired or the data whichever is needed, it should not be given any disclosure. So it should be kept confidential. Those data might be, the patient may be a cancer patient, he is involved in a cancer study, his preliminary data or the post data, everything, nothing to be revealed outside. So because it is a valid legal document, it is considered as a legal document, it should not be produced unless otherwise it is required or requested to do so. Maybe of any scientific reason also it should not be given. And the principles of precaution and risk minimization. So this due care and the caution to be taken at all stages of research from its inception, maybe as a research idea if it, when it is inception, that is during its conception stage also, and its subsequent research design and the conduct of research or experiment and its applications, everything will be due care and caution to be taken in the all stages of research, starting from the inception till the delivery. So how far we'll be treating a that is a conceived person, so how far we'll be taking care. So those due care and the caution and the attention to be given to the that is to the all stages of research also. So this to ensure that the research subjects are put to a minimum risk and they suffer from no irreversible adverse effects. That is very, very important, because after taking this medicine, the person should not have a permanent hepatotoxicity or permanent loss of vision or permanent loss of any senses. So that no irreversible adverse effects. For example, streptomycin, this aminoglycosides are a hearing loss will be there. In some of the chloroquine, there may be vision impairment will be there. So those drugs, it should not, that is while screening upon the drugs upon the clinical patients, they would have come across those adverse effects. When it is being given to a larger population, it should be an irreversible, it should not be irreversible, it should be reversible. So principles of professional competency. So the research is conducted at all times by competent and qualified persons who act with total integrity and impartiality. So all should work for a noble cause. Supposingly, for if we are assigned a task, so we'll be constituting a team who will be having the mindset that is called as the like-minded people. So those like-minded people to associate together with a total integrity and impartiality to make the mission a success. So this research is conducted in all times by a competent authority, that is a responsible. Competent is a responsible authority and a qualified person. So everybody cannot come and take the blood sample. A nutritionist should come and design how the diet should be. The physician should come and see that how frequently the drug to be given, and then the lab technician to come to withdraw the blood samples. So like that, there should be the qualified persons and competency should be there with the total integrity and impartiality, and who have been made aware of. So preliminary, there should be a discussion. They should have been made aware of. They are mindful of the ethical considerations to be borne in mind in respect of such research or experiment. So the ethical considerations to be borne in mind in respect of all the research or experiment. So principles of accountability and transparency. The researcher should conduct the experiments in fair and in a honest way, impartial way, transparent manner. Anywhere the details are to be asked, so it should be very transparent after full disclosure of his or her interest in the research. So they should also retain the research data subject to the principles of privacy and confidentiality for a minimum period of 5 years. For a minimum period of 5 years, the research data and the subjects who and all are involved. So all the details to be kept very confidentially for a minimum period of 5 years to be scrutinized after it has been scrutinized by the legal as well as the administrative authority if necessary. So even for the NAC or else for the NBA purpose, we maintain the data for at least 5 years, the last 5-year data. Likewise here also, the accountability is very important and the transparency is very important. The raw data are very, very important. There should not be any mismatches between the before and after the statistical calculations. Next is the principles of the maximization of the public interest and of distributive justice. So the results of the research should be used for the benefit of all humans. So this is of course the first in my first slide I said about the ethics, that is about the whether it is going to be benefited for the society or not. It should be used for the benefit of all humans. In particular, the research participants themselves or for the community, maybe of the cancer affected community, or else maybe for the neurological patients, or else an arthritis community, or it should be based benefit a community from which they are drawn, and not only to those who are socially better off. There should not be a disease which can be like a what I say is like a lifestyle modification, obesity. For that and all, we cannot go for a clinical trial, not for a socially well off. It should be given to it should be suitable, and it should be made eligible and made more sensitive to the patients who are not socially well off or maybe of a weak economic status. Those patients also should get benefited out of the treatment. So, for example, it should be a very prevalent disease which is affecting all sort of populations of the community. That is called as a distributive justice. So, principles of institutional arrangements. So, it is required that all the institutional arrangements required to be made in respect of the research and its subsequent use or application should be duly made in a transparent manner. So whatever the arrangements we have to make it up, so everything to be given in a transparent manner. Take all the appropriate steps to ensure that research reports, materials and data connected with the research are duly preserved and archived. So that is, it is being saved. So it should be everything to be done in a transparent manner. So principles of public domain. So the principle of public domain, that is the results of any research work done should be made public through the publications or any other means. Whenever they go for any generalized studies, those studies to be given in the form of a publication so that it can be reviewed by some of the reviewers, to be given some comments, and then only it will be known that whether it is suitable for the population or not. Even before publication, the detailed information of the clinical trial should be made public before start of the recruitment via the clinical trial registry system, because they have the clinical trial registry system. Through that only the volunteer goes for registration. In that registration itself, the every information should be made available. Principles of totality of responsibility. So all those who are directly or indirectly connected with the research should take the professional and moral responsibility. You should not say that I didn't withdraw blood from the participant by that time. It is he who had made it, and I haven't given food to that candidate for that particular time. It is that another person had done it. It is not of that category. This is the totality of the responsibility. So if the team comprises of 100 members, all the 100 members are responsible for the success, or even if there are any misleads or any of the failures in this thing or any risk associated, everybody should take the professional as well as the moral responsibility for the due observance of all the principles. So that you should not say that I am not there in that. If you are there, if you were there in the place, how it would have happened. So the same manner should be repeated with all. That is of all the principle guidelines, the prescriptions laid down in the respect of the research. So it might be of the researchers, the funding agency, institution where the research is conducted. So these category people are responsible. This is the principles of totality of the responsibility. The persons, groups, or the group of undertakings are who sponsor the project or the study, and who are used to derive benefit from the research, and who are to market the product. Any companies are going to market the product, they are conducting the trials. Or if I am the lead compound manufacturer, whether I am going to get benefit out of the research, or else I have approached a sponsor, that sponsor. So those, or else the involved persons, everybody will be responsible for that too. Principles of compliance. So this is very important, that the all those associated with the research work should comply by the guidelines. Whether you are going to follow the ICH guidelines or whatever the guidelines, that guidelines to be followed very strictly, pertaining to the specific area of the research, to that particular area. So whichever guidelines are very important, so that has to be made, that has to be, surely it has to be followed with. For research to be conducted ethically, we need to follow all these 12 principles of the principles laid down by the ICMR. So in order to follow these principles, we should be aware about the informed consent process also. So it is mandatory that all the proposals in the biomedical research should be cleared by the appropriately constituted independent ethics committee, that is IEC, or the institutional review board. So this institutional ethical committee, the basic responsibility is to protect the dignity, rights, and well-being of the research participants, to ensure universal ethical values and international standards which are to be followed, to assist in the development and education of research community responsive to local healthcare requirements. This is the composition of IEC. So they may be multidisciplinary and multisectorial. First person will be the chairperson, one chairperson, 1 to 2 basic medical scientists, 1 to 2 clinicians, 1 legal expert, 1 social scientist, and 1 philosopher, ethicist, or a theologian, and 1 lay person from community, and 1 member secretary. Maximum members are 12 to 15. Minimum quorum is 5 to 7 to conduct an institutional ethical committee meeting. So this is about the ethics, what we need to follow in the clinical research. Thank you all.
Ma'am, thank you so much for your very informative session. And also you clearly explained the use of ethical principles and their need for consideration during designing of clinical research. There are a couple of questions, ma'am. Is informed form required when we collect only data from participants?
What is it?
Is informed consent form required when we collect only data from participants?
Yes, we need to take it, because we need to say them that what for this data we are taking it. Might be there, if for example, if you are taking any data to be compared before this treatment and after this treatment, the patient should be made aware that why we are taking it.
Thank you, ma'am. There's one question, ma'am, that is from last session. What is the further condition of volunteers if any lethal effects were observed?
That's what they have to be under medical insurance. So that insurance cover, if some untoward event or incident has happened, then the members will be given the dues, because they are giving the acceptance, then only they are just getting included in the study. If the fate is like that, then the family members will be given the insurance coverage, whatever they have covered in them.
One more question from current session, ma'am. Does the ethical guidelines cover the long-term effects of drugs, like after 5 years?
After 5 years, it won't consider. So only that is up to 5 years, whatever the effects are happening, so that will be held responsible. So because we have the data up to 5 years, after 5 years we are not they not, but there is some consideration that there may not be 100%, but the percentage variations will be there for if it is after 5 years.
Are there any still questions from participants?
One question in the chat box again.
Yes, ma'am. I did read it. I just read it. Okay, ma'am. Thank you. Asha, ma'am, you can.
Yes. Thank you, Shreya ma'am. Thank you very much, Dr. Chitra ma'am. We are very
Yes, thank you.
grateful for your time and energy what you have put forward for the sessions. I request Dr. Vinay sir to showcase the certificate of appreciation which we are presenting to the speaker of the day.
Yeah, just a
Can I
minute,
stop
madam.
share? I'll stop sharing.
Yes, ma'am.
Yes, ma'am. So, dear delegates, as we are approaching to the end of the session, we are not yet closed. So, please be patient for 5 more minutes. We'll just have a little showcase of the certificate of appreciation to our guest speaker, followed by the virtual souvenir release for 2 minutes, which will be the hard copies of the souvenir will be distributed on the offline days. So we'll be having just a virtual release of the souvenir after the certificate distribution.
Just a minute, madam.
Yes, sir.
Yeah, I'm very thankful to Dr. Chitra madam, today's resource person, for spending her valuable time. So thank you very much, madam, for accepting our
Thank you, sir.
request. For accepting our
Thank you,
request.
Dr. Vinay.
Yes, madam. So, dear participants, as a token of gratitude to the today's resource person, so herewith we are herewith we are sharing the presentation certificate to the resource person. Madam, please accept our token of
Yes.
gratitude, madam.
Yeah, thank you, Dr. Vinay, and the convener for giving us the opportunity to share my views in the ATAL FDP program. So I'm very happy to be a part of this.
Yes, madam. Madam, is it visible, madam?
Yes, visible.
Yes, sir.
Madam.
Yes.
Yes, madam. Yes, madam.
Thank
Madam,
you very much for sharing. Yeah.
Yes, madam. Thank you, madam. Madam, soon we'll send the certificate to post.
Okay, sir. Okay. No issues.
Okay, madam.
Thank you, doctor, and thank you all the participants as well as the organizers for making this event a successful one. And I'm very happy that I could see again the same 40 participants in the first session and the second session also. This shows their interest towards this FDP. So I wish the FDP a grand success. Thanks for the opportunity given to the organizing secretary and to the convener. Convey my regards to your management also. Thank you.
Thank you very much, madam.
Thank you very much, madam.
Dear participants, please don't leave the meeting just for a few minutes. We'll share the souvenir inaugural virtual souvenir inaugural session. So, dear participant, actually, at the starting of the session, just I told about the instructions of the FDP. Just whoever missed the session, just I am giving brief lines about the FDP. As you know that this is hybrid FDP, you should attend online as well as offline sessions. And in the offline sessions, you will be having the handling of softwares and one workshop and also one industrial visit. So hope you will attend the offline sessions also, which are commencing from 12th of this month. And lunch and high tea and also transportation to the industrial visits will be provided from the management. And also one more thing, as the FDP is outcome based FDP, so as per their guidelines, they're giving the certificate those who clear the assessment, that is MCQ exam. So that MCQ exam will be conducted this Saturday. So that is on the 10th of this month through online mode. So one first MCQ exam and the second MCQ exam will be conducted through offline mode at our college. So those who are participating for online mode, those they need to come to the college on the day of assessment, and that offline assessment will be conducted on the Friday of the next week. That is on the 16th September 2022. Offline MCQ assessment will be conducted at our institute. So the participant has to appear for both assessments, both MCQ exams, in order to get the certificates. So still, if you have any doubts, you can please contact me. My number is there in the ATAL FDP participant group. If you have any difficulties and if you have any queries regarding this. So now I would like to share the inaugural session of the souvenir, so which will be handed over to the participants during the offline session. Whenever you are visiting our campus on the Monday, coming Monday, 12th of this month, so we will be distributing the souvenirs and also some these PPT slides, and also softwares also we are going to demonstrate. And if you are interested, we will also install the softwares for you, like clinical SaaS, CDM softwares like that. Right. So, madam, can I share the
Yes, sir.
virtual release of this one, madam? Savani?
Yes, sir.
Yeah, yeah.
Yes,
Okay,
sir. Meanwhile, after this, sir, we'll be sharing the attendance and feedback link.
yes, madam. Yes, madam. So, is it visible, madam?
Yes, sir.
So that's it.
Yes, sir.
Okay, dear participant. So now I am sharing the attendance form, come registration form. Please fill up the details, your mail ID, your phone number, designation, and your organization will be there. Please fill up the details. The same thing will forward to the ATAL website. And also feedback form also I am posting in the chat box. Just now I posted the feedback form. So you can find out the feedback form in the chat box. And also I just I am just now I posted this one also, registration and attendance form also. The first one is feedback form, and second one is attendance form. So you need to click on both the Google Forms. So please share your feedback regarding the first day session, and also please mark your attendance by using the registration cum attendance form. So dear participants, hope you understand all the session details. Today 2 sessions we have conducted, and while tomorrow also we'll meet by 7 o'clock. So today I think this is the first day, that's what it got delayed little bit. So tomorrow exactly by 6:45 I'll be sharing the link. And one more thing I would like to bring it to your kind notice, that the same Zoom meeting link will be you can use for all the 6 sessions. Same link, same user ID, and password is also same. You can use the same credentials for all the 6 sessions of online sessions. Yeah. And any feedbacks from participant side? Any feedback from participant side?
Yes, we request the delegates to come forward with any suggestions or any queries you have. So we are getting replies in the chat box. Thank you very much to everyone who all are responding in the chat box. If anybody else would like to go for any queries or suggestions, you can drop directly a message to the coordinator. Sir will respond to you.
Sir, okay. Sir, now I would like to request our principal, sir, to just give the closing remarks for this day. Sir, I would like to request the convener of the program to give the closing remarks for this today's presentation.
Yeah, Dr. Vinay sir, thank you very much for the well conductance of today's first session by Professor Chitra Madam, Dean SRM University. So we have, I think, all the fellow faculty members, you have received a valuable information and useful information from Madam. So I could see the number also. So you almost continued. Madam also said the same. More than 40 members, even first session and second session, both the sessions, participants also is there. It is a good sign to all the faculty members. So it is a good sign to the faculty. So you are coming forward to learn and to acquire knowledge for your upgradation of your professionalism. So, and once again, I congratulate the team, ATAL FDP hosting team, the pharmacology and pharmacy practice department. So for your hard work and the in time, the conduct of the program and in time the completion of the program. So I hope in upcoming sessions also you will be giving the same. You will be continuing the same. So thank you, Vinay sir, and thank you,
sir.
Thank you, sir, for your closing remarks. Sir, Vinay
Yes,
sir?
yes, madam. Dear participants, you can sign off. You can log out. So tomorrow by 6:45, you can join the session. So once again, happy Teachers' Day to you all. Thank you very much.
Thank you, sir. Good night, one and all. We'll see tomorrow again. Signing off for today. Good night.